Okay, well, let's go ahead and get started. Well, hello, everyone, and welcome to another exciting edition of the CompTalk Communities of Practice. My name is Sammy Hamp, and I am a Public Affairs Specialist in the Center for Computational Toxicology and Exposure. Today, we'll be hearing from Dr. Carlie LaLone about SeqAPASS version 5.0. And before we get started, I just want to make sure that everyone please mutes their lines. You can accomplish that by hitting #6. And then without any further ado, I will hand it over to Dr. LaLone.
Great. Well, thank you, Sammy. And hello, everybody. And hope everyone's having a happy holiday season. Really excited to be able to talk to you all today. It seems that our SeqAPASS cycle, the cycle in which we release the sequence alignment to predict across species susceptibility tool, falls in line with this Thanksgiving holiday season. And so it's really nice to be able to get an opportunity to always come to this communities of practice and share the new features with you all. So recently, actually December 1st in the afternoon, we released version 5.0 to the public. And this round of development over the last year has really focused on suggested features from you all, so from the user community that are really working with the SeqAPASS tool for their various research purposes as well as decision-making purposes, and communicating back to our team either at professional meetings or via email of features that would be useful for really thinking about evaluating the data and really rapid data synthesis and interpretation. So that was really our major focus this year, is bringing forward the features that can help with interpretation of the data. And so to start off, many of us who attend these CompTalks communities of practice, we think about chemical safety. And we know that in our modern world, chemicals make up almost everything we see and use. And really to keep pace with this modern lifestyle, we need to be developing, and there are chemistries as well as chemicals continuously being developed. And as researchers, we're at EPA, our mission is to protect human health and the environment. We're interested in the effect of these chemicals both to human health, but then certainly across species as well. And so this is a pretty complicated issue when we're thinking about understanding from available information that we might have about surrogate species or model organisms in the literature, or that we're generating data for in the laboratory. When we have certain existing information for those specific species, how might we be able to utilize that information to extrapolate across species and have some understanding of possibility of effects across species? And so, like I mentioned, it's a complicated issue, right? There's the toxicokinetics and the exposure and thinking about different life stages of organisms and life histories and their exposure, as well as absorption, distribution, metabolism, elimination. And then also there's the toxicodynamic side and understanding the biological pathway and how a chemical may interact with a biomolecule that then initiates this cascade where we might understand some of the different impacts on different levels of the biological pathway that may culminate in an adverse outcome. And much of our work has really focused on utilization of techniques in bioinformatics and how we might be able to take advantage of some tools in this arena to be able to understand species similarities and differences. And so our major focus has been on proteins and understanding protein similarity and differences, so kind of at the molecular level here. And so just some very, very basic biology on amino acids and proteins in general. Amino acids are encoded in genes. They come together to form this polypeptide string. A lot of times it's depicted as a pearls on a string, and these amino acids then come together to fold into both a secondary structure and then this tertiary structure. And that tertiary structure is really important for the function of that protein in the body. And specifically, there's an interest in understanding how natural ligands, first of all, as well as chemicals, can interact with that protein structure. So we can have an interaction in a specific pocket in a protein And that could be at a functional domain. So just thinking about nuclear receptors as an example, chemicals or natural ligands interact in specific pockets that might be their ligand binding domain. Also, that chemical protein interaction may be understood as far as critical individual amino acids, like we talked about those pearls on a string. Certain amino acids are very important for maintaining the structure of that protein, as well as for maybe even directly interacting with a chemical or even a natural ligand, either through hydrogen bonds, for example. And as those chemicals interact with a protein, say at a molecular initiating event in an adverse outcome pathway, it gives us an understanding of perhaps what types of effects we may be able to see based on that direct interaction if we have knowledge of an adverse outcome pathway. So what this work has really focused on is thinking about similarity across species at the molecular level of the protein. And for kind of predictive approaches that center around computational approaches to understanding this similarity, we're really thinking about conservation and looking at, sequence as well as structure, and ultimately we want to be able to be predictive of potential function of that protein in other organisms. And really, as bioinformatic platforms evolve, we hope to continue to move forward with both sequence, structure, and then further into function. So for now, the work that we've focused on has taken into account available protein sequence information and different information that we can glean from structural knowledge of a protein. So what we can do is, in thinking about cross-species extrapolation, is if we have an understanding of how a chemical interacts with a specific species, and we know that it interacts with a particular protein, say at a molecular initiating event, and in this example, thinking maybe the rat would be that species that we have information about, then we can take that information and we can start comparing sequences, and we can compare to hundreds and thousands or thousands of species and understand how similar, how well conserved that protein is as an initial step to predict whether or not a chemical could be similarly acting in other species. And so that's exactly what the sequence alignment to predict a cross-species susceptibility tool was designed to do, is really gather lines of evidence towards conservation to help us predict susceptibility to chemicals. And like I mentioned, with each iteration of this tool, we really built it, custom built it for the users and tried to make sure that many of our interactions with both internally within EPA, but also externally, are helping to drive the features that we're developing each year. So just to talk about the SeqAPASS evaluation, we wanted to make it very flexible based on how much information is available about a chemical protein interaction, right? So, the very most basic level of comparison is looking at that primary amino acid sequence and doing alignments at that level. So level 1 of the SeqAPASS tool in this streamlined process is comparing the full primary amino acid sequences across species, and then making a call of whether or not we think that protein is conserved or not conserved. The next level is, as we talked about, when those proteins fold up, there's functional domains that are important for being able to have the desired capabilities that protein needs. And so we talked about the ligand binding domain. Certainly there's others. There might be a domain that's involved in the enzymatic reaction for an enzyme, or DNA binding domains, for example. So we can gather information on what we know about functional domains within a protein, and we can compare those specific functional domains across species. And then finally, if there's information available on that interaction with critical individual amino acid residues or close contact amino acid residues, we can look specifically at those individual amino acid residues across species. And what we're doing at each level is really gathering lines of evidence toward, protein conservation, so that ultimately we can say whether or not we think, our rat species where we have information and understand the interaction, whether or not it's likely that fish species, for example, also contains that protein, and that chemical could possibly act in a similar manner because at each level we're seeing conservation, or at least at each level that we're able to perform the evaluation based on existing knowledge, we see conservation of that protein target. So it's a very initial question that we can ask, is that protein present or not? And so that's exactly what the prediction and the output from SeqAPASS looks like, is basically we're predicting the likelihood of similar susceptibility based on that sequence conservation at those various levels. And so the lines of evidence will just indicate a yes or a no across the species where we have sequence information based on whether or not the protein is conserved or whether or not it's not likely to be conserved. So I think we need to be very clear of what SeqAPASS does and what it currently does not predict, right? So we need to be clear that SeqAPASS currently is a yes or no prediction of conservation of that target across many species that can be performed very rapidly. And it does not predict the degree of sensitivity or susceptibility. So the percent similarity, for example, is a common metric for understanding similarity of alignments. And that percent similarity doesn't necessarily equate to the degree of susceptibility. And that's why we're making a call of susceptible yes or susceptible no. And so as we develop the SeqAPASS tool, and each time we have a new iteration, we're trying to roll these new features out to the public. And we're trying to demonstrate how we're taking advantage of existing and available information on proteins and chemical interactions, and keep developing this very science-based, streamlined, and transparent, publicly accessible pipeline that takes advantage of as much sequence information, as much structural information as we possibly can to be able to predict susceptibility across species. So to do this, we look for people that are interested in enhancing their extrapolation capabilities. So a lot of times champions and organizations that recognize that this tool can be applicable to many of the research questions or decision-making questions that they might have. And as a team, our group specifically, my team develops case examples to try and demonstrate different types of applications of the SeqAPASS tool for both decision-making purposes as well as guiding research and using it as a hypothesis generation tool besides a extrapolation tool. And then work to provide the training for the use of the tool. Maybe many people aren't necessarily well-versed in bioinformatics or haven't really worked much with sequence comparisons, but that shouldn't inhibit them from using this tool because our objective was to develop this tool so that it could be useful for anyone with a basic biology background, and providing the training to make sure that that's consistent. And then further, when people are developing new case examples, we try and provide the support that they might need for their evaluation, particularly as new users begin. So we always are able to gain insights from, on the development standpoint, from these types of interactions through developing case studies, through training both internal and external partners, as well as through supporting people that are utilizing the tool for the first time or a new case example, for example. All right, so with training, we go through and we talk about this is how you set up a level 1, a level 2, and a level 3 evaluation, and here's the type of data that you get. And a lot of times we have people looking at us like, okay, yes, we got this, we can take it back to our offices and start working with the tool. And so something that we want to avoid, of course, is once we get people working with the tool by themselves, we don't want them overwhelmed by the output or the input even of how they get from point A to point B as far as what information goes in and how do you interpret the data coming out. And so our focus is really on this rapid data interpretation this year through data visualization and then customizable and transparent summary reports. And so I'm going to talk about the new features that have been added to SeqAPASS. But first of all, just kind of starting out and explaining what type of data comes out of the SeqAPASS tool and how it's presented to the users. So as I had mentioned, for level 1, where we're looking at the full primary amino acid sequence, as well as level 2, where we're looking at functional domains, ultimately, we're getting a prediction of susceptibility of yes or no. So across, like I said, hundreds or thousands of species very rapidly, you get information on those species as well as the proteins that are aligning. There's different metrics associated with that alignment that's transparently presented in the output. And then you get that prediction of susceptible yes and susceptible no in these large table Excel or CSV files that you can look at and understand that data. However, Excel sheets aren't for everybody. And so a very rapid way to synthesize and understand the data is to put it in a data visualization. So those of you that are familiar with version 4.0 and a little bit earlier than that, we had developed these box plots. And this is a way to very rapidly evaluate the data that's presented in SeqAPASS level 1, where we're looking at primary amino acid sequences, and level 2 functional domains. So here we have the different taxonomic groups listed on the x-axis, and then on the y-axis is the percent similarity compared to this query species sequence here. So this, in our example, if we just carry that forward, if we're looking at a rat sequence and comparing to all other species that have sequence information available, we can rapidly look at this data and understand that based on the susceptibility cutoff that's generated through identification of the lowest percent similarity in the data set, where we're still identifying ortholog candidates that are more likely to have similar function, we predict that out of these species that we can align, which species are more likely to be susceptible that cross that, which taxonomic groups, at this level, are more likely to be susceptible, and those that are less likely to be susceptible that aren't crossing that line. So here, very rapidly, we have a prediction for over 1,000 species. And then if we want to get into more detail, we have some summary tables that if you click on a box, so here if I click on the Actopinuri, which is bony fish, we can very rapidly see that there was 202 species from that taxonomic group that aligned with the rat sequence. And we can rapidly get the median and mean percent similarity as well as the prediction of susceptibility across that taxonomic group. So this is what the level 2 as well as the level 1 visualization looks like. So for version 5, we really wanted to bring forth a rapid way of evaluating the critical individual amino acid residue comparisons. So just like level 1 and 2, we have table output that can be downloaded as CSV and Excel files, and certainly you can mine it within the tool itself and look through those tables. And ultimately, there's a susceptibility prediction or a call on susceptibility based on looking at side chain classification of that amino acid residue and similarity in that side chain classification, as well as using molecular weight as a surrogate for size. So, massive changes in the size of the amino acid as well as differences in that side chain classification are more likely to lead to differences in an interaction with a chemical than those that are more or less severe, I should say, or less of a change, or maybe they're exactly matching that of the query or the template sequence. So ultimately, we present all of the data demonstrating, what position the amino acid that aligned is in other species, as well as, what is that? Here it's an abbreviation for amino acids. But what we do is we try and present that data very transparently in large tables. But sometimes, again, that can be relatively overwhelming to look across and rapidly understand the similarity predictions. So therefore, I'm just going to chat through the SeqAPASS setup for aligning amino acids in the tool. So here's just a screenshot of level 3 in the SeqAPASS tool, where we've chosen to use the human sequence for androgen receptor as our template sequence in which we'll compare all other sequences that we can customize this alignment for critical individual amino acid residues. Here we chose to look at mammals, turtles, as well as bony fish and make some alignments. And then from the literature, there was information from human species on which amino acid residues are critical for interaction with a specific chemical. And so we set up this alignment and we generate those tables that I had just shown you. However, now we have this drop down here where you can click in and generate a visualization of that data. So to access the heat map, this window pops up, and you can click on the heat map visualization, and it brings you to a customization form or area to be able to customize your heat map. And in the tool itself, the heat map would be seen below this controls area. But just to break it up and be able to describe it a little bit more thoroughly to you in the presentation, you get to choose which taxonomic groups you'll be comparing in your heat map. You can customize it from a simple report to a full report. So a simple report might only report or only reports the position of the amino acid and the amino acid itself, whereas the full report will give you all of the additional information on side chain classification as well as molecular weight, for example. Change the scientific to scientific name or common name. And then there's these specialty categories that you can add into the data. So you can choose to display the ortholog candidates or highlight them. You know, those of you interested in threatened and endangered species, you can readily pull that information out of the data and highlight that in the heat map. Or say you're interested in where is the common model organisms fall out, you can also click on this radio button, and that will be presented in your heat map. And then below there's these heat map settings where you can choose to display the text in the visualization. So I can show what that means later, but you'll be able to see that there's different ways that you can customize the visualization for whatever purposes you have, whether it's for just rapidly synthesizing the data or presenting the data, communicating the data, or even putting it in publications. So here's what the heat map looks like with all of the text on it. But what you can see is we have whether or not it's a match or not a match, and you can rapidly pick that out from color, and then we have susceptibility predictions. So basically these heat maps allow you to very rapidly understand similarities or differences within the data set. And it also lets you look at the susceptibility predictions. As I had mentioned, there's specialty groups that you can highlight. So here, highlighting endangered species, and it's an interactive visualization in that if you're hovering over different species or even in the amino acid area, it brings up additional information. So for example, we know that for this species was a predicted sequence that had aligned to the template sequence. And then it also gives different categories that are also aligned out here. So, it's also an ortholog candidate, and it's also a common model organism. Further, like I mentioned, you can remove the text for the visualization. You can also customize so that you remove all the amino acids and only have the susceptibility column, or you remove the susceptibility column and only have the amino acids. So there's a lot of customization that can occur, and like right here, we're showing that you can also click and have the ortholog candidates displayed and highlighted on your heat map as well. So I would encourage everybody, I couldn't get through all the different types of features that are available through that visualization, but to go check out the SeqAPASS tool itself at seqapass.epa.gov/SeqAPASS. And we're really working to move from an overwhelming amount of information to something that's very readily understandable by our users for their specific queries and purposes of utilizing the tool. All right, so another feature that was integrated in the SeqAPASS version 5.0 is that of a decision summary report. So in earlier versions of the SeqAPASS tool, the user could go ahead and they could, pull out each piece of information. So we talked about tables at each level, we talked about visualizations at each level, and so those could be downloaded as in different formats, and the user could stitch those different predictions from each level together. But what we suggestions from our users and just kind of understanding what types of data are presented, we wanted to be able to bring that information all together in a single report that could be downloaded as a PDF and would contain all of the information that somebody might want for their reason for querying the SeqAPASS tool. And so what we did is we integrated some integrated capabilities to be able to push tables and push the box plot visualizations and the heat map to this decision summary report. So therefore, when you're looking at level 1, you can push the table from the decision summary to the decision summary report, or level 2, level 3, etc. And then further, there's buttons on each page to be able to push that data. So on each page itself, when you're working, you can customize that table so that you can view what you'd like to view, and then push it on over to this decision summary report. So what that looks like then is, here's the different buttons if you're familiar with working with SeqAPASS for the different levels where you can, look at the tables and visualization for 1 and 2 and 3. But once you push information using those buttons on the different pages to the decision summary report, this button up top that says DS report becomes active. And when you click on it, it'll bring you to a customization area to be able to set up your table that you can output, as well as the PDF that I had mentioned. So here you can select which taxonomic groups you'd like incorporated into your output. And then, you can also go through and be very specific about which species you'd be interested in including, and then you can, change that among the common and scientific name. You have all of your query information available up top here that can be integrated into your PDF so that you all have all of that information of what was run. And then there's this area here, this optional components, where you can include in your PDF both so this part lets you have the table, but if you do click on these different selection buttons, checkboxes, you can include this query information as well as the visualization, or you can deselect that and not have the visualization. So again, very customizable for level 1. For level 2, as I had mentioned, with level 2, we could be interested in multiple functional domains. And so that's why it's presented a little bit differently, because here I ran only one ligand binding domain for the androgen receptor. But say for another protein, maybe I had interest in multiple domains, they would all be listed in rows below here. And then you could choose to add it to the report table that's actually below, or you can also click to add this this type of query information and the visualization that you've already pushed to the decision summary report to your PDF. So that's another selection option. And then for level 3, so we have the query information. It tells you what your template sequence and species that were used, as well as the selected individual amino acid residues at their position, and then the abbreviation for that amino acid. And then you can choose to add this to the report below, add the information as well as the visualization. So it's really customizable as to how you set this up. And this is located below those control panels. So as you're selecting what you'd like to incorporate, you get that information displayed below. So here's the level 1, level 2, and the level 3 template, as well as the susceptibility prediction across the various species in one area. So you can download that as a CSV or as an Excel. So it's very rapidly, you have that information for each level of the analyses customized for the species that you'd like, or the taxonomic groups that you'd like, readily available there. And then further, like I mentioned, we're working on creating a decision summary report that comes as a PDF. So if you click on this button here, it brings up the decision summary report. So we're going to see how quickly this loads, because I do have myself and my 2 kiddos accessing our Wi-Fi at the same time here. So here we go. We have the decision summary report. And what that has is, so I've pushed the level 1 query information, as well as, oops, I'm sorry, I didn't mean to click so quickly, as well as the level 1 visualization. Here's level 2, as well as the level 2 visualization. And then we have the level 3 query information, followed by the heat map visualization, and then further the final decision summary report table that presents all of the information that we saw in the previous table, level 1, level 2, and level 3. So that's a downloadable PDF report that you'd have generated for that specific evaluation that you completed. So just to talk a little bit about the evolution of the SeqAPASS tool. So, we first initially developed the functional version of doing the evaluation of level 1 and level 2, which had pretty much the initial essential functionality in 2016 and released a publication focused on that with case examples. And then in 2017, we talked about how you can compare critical individual amino acid residues. And in 2018, then we developed visualization for level 1 and level 2, as well as we automated the pipeline for making those susceptibility predictions using level 3. And then again, in 2019, we worked hard to try and improve visualization options, as well as created a guidance document, a user guide. So that's very thorough. So those of you that are new to SeqAPASS, we have a lot of information buttons integrated within the tool so that you can rapidly, learn the tool as you go, but also if you have more in-depth questions or something just isn't functioning the way that you think it would, there is a very visual user guide that has a lot of screenshots and text description of how the tool works. There's summary tables as well that we talked about, as well as interoperability was a big piece of 2019. So the tool also, it gives predictions based on these protein comparisons, but it was also desirable to link to available empirical data that either, that could support your evaluation. And so we added a feature where we can link out to the EcoTox knowledge base, which captures single chemical toxicity information across a wildlife species. And so that's a curated database that we can link our predictions of susceptibility to empirical data when it exists in both our predictions on SeqAPASS as well as in the EcoTox knowledge base. And then further, there's links to the CompTox chemicals dashboard, which has a great deal of both physiochemical information as well as toxicity data, for example, and bioactivity data based on in vitro assays, and the adverse outcome pathway wiki. So that was really important to be able to have some of that interoperability, because again, in that data synthesis, especially for our decision makers that are really being asked to use many different types of tools, this type of integration can be very useful for helping them grab the information that makes the most sense to be placed together very rapidly. And so that's something that we think a lot about at EPA, is how we can be more interoperable with other tools to be able to bring the most relevant data together for specific uses. All right, and so then with version 5 here in our development team, I hope you do check it out. We have the visualization, the customizable visualization for level 3, and then we developed this decision summary report that again was really designed and developed to bring together the different levels of the SeqAPASS evaluation in one place that can be saved and downloaded and used as both a report as well as, for your presentation communication needs. So with that, I'm really happy to be able to be kind of the voice of SeqAPASS and present our tool as it evolves. And, one thing that I want to mention is, there's a much larger team that's behind the scenes and working a lot to make this tool successful as far as developing the case examples. So we have Donovan Blatz, who's an ORISE fellow, who works with me in doing a lot of the user guide development, the standard operating procedure for testing the tool, as well as creating case examples of how you utilize the data. So currently he's interested in cytochrome P450 enzymes and metabolism in different types of bee species, and using SeqAPASS as a means to evaluate differences in ability to metabolize different pesticides. And then there's Sarah Vliet. She's a postdoc that's working with me, and she's interested in application of the SeqAPASS tool for the androgen receptor and transthyretin, so different types of endocrine targets, and then incorporating SeqAPASS with systematic review opportunities to bring together a pathway-based evaluation of the androgen receptor pathway to define the domain of applicability for across species. So that's a really nice case example demonstrating the utility of SeqAPASS. And then Sally Mayasich is another postdoc that we're really working to keep up with advances in bioinformatics. And so Sally's on our team, and she's working on thinking about how we can advance beyond our sequence evaluations and bring in more molecular modeling across. So when we have say we have an X-ray crystallography structure available to us for a specific species, how we might be able to use that to generate models for representing many species from the output of SeqAPASS and advance that into molecular docking, for example, and how that would enhance our ability to move from a yes/no susceptibility prediction to more in-depth and maybe even a rank order of those types of species that may be more susceptible or less susceptible. So that's something that Sally's been thinking a lot about. And then Marissa Jensen, she's very interested in adverse outcome pathway framework and being able to utilize the SeqAPASS tool to basically walk down an adverse outcome pathway where there's proteins involved both in the molecular initiating event and early key events. How can we utilize the SeqAPASS tool along with other relevant in vitro and in vivo information to define the domain of applicability for adverse outcome pathways? And so that's my team that's really working on application of the SeqAPASS tool, and as well as site-directed mutagenesis studies, so that not only can we have these predictions of susceptibility based on changes in individual amino acid residues, but then using the laboratory to confirm those results and see that when you do have a change in an amino acid residue across a species that we're predicting to be, either sensitive or not sensitive, do we actually see that in the laboratory? And then there's the GDIT team that develops the SeqAPASS tool. So Tom Transu, Cody Simmons, and Audrey Wilkinson. They are the nuts and bolts behind the development and the release of the SeqAPASS tool. And so with that, I'd be happy to, I hope you got a sense of what those features look like that we've added to the SeqAPASS tool. And if you're interested in utilizing it for, applying it to some of your research or decision-making needs, I invite you to reach out to myself. My email is there, or my team, and we'd be happy to get you going with the SeqAPASS tool. So I guess I will end there and see if anyone has any questions for me.
Cool, yeah, it looks like we do already have one question in the chat from Sergio Villalobos. Do you want me to read that out?
My way back.
You can hear yourself, Carlie. Cool.
All right, could SeqAPASS be used to better understand processes such as bioaccumulation across aquatic animals? Well, yes. So that is an interesting concept, right? So bioaccumulation, something that we've started thinking about more so this year than any other year, and we have some publications coming out on this with Carla Ng out of the University of Pittsburgh, as well as Hui Peng, is how SeqAPASS can be used to predict bioaccumulation potential of perfluoroalkyl substances, or PFAS, because of the importance of protein binding. And so there's certainly ways that when we have some understanding of bioaccumulation relative to protein interaction, where understanding that conservation could help us in understanding bioaccumulation. So yes. And then at issue is the regulation is based on bioconcentration factor in fish, but fish have enzymatic machinery that invertebrates such as mollusks and crustaceans do not have. So if bioaccumulation is the end result of an organism's ability to dilute or concentrate a chemical, and fat or protein storage is critical due to affinity principles, could the advances on this tool provide insight? Absolutely. So even with the cytochrome P450s, right? So if we understand that this, we know that this is a primary metabolizing enzyme that's important for either the activation of this chemical that leads to toxicity or leads to a lack of toxicity. So if we have some sort of understanding of the enzyme itself and how it performs in a species, we can utilize that information to understand whether or not that target is conserved in other species, that cytochrome P450 is conserved in other species, and that would give us some advances and some insight on bioaccumulation. Yep. So I think that's a really great point, and that's certainly something that we've, just the evolution of the SeqAPASS tool has been really focused on chemical susceptibility, but that's a broader question beyond just the target interaction. It could be, that those important metabolizing enzymes as well, or even, like binding proteins if we're thinking about, like I said, PFAS. Can you consider combined effects, multiple toxicants rather than one at a time? Synergistic effects are important parameter to consider. So definitely, but so, okay, at level 1 and level 2, though we're thinking about perhaps chemical susceptibility, it's really at level 3 where you're comparing those critical individual amino acids where this tool becomes very chemical specific. So you could compare multiple toxicants at a time if you knew that they had the exact same interaction with that protein. Then yes, they could be grouped as a group that interacts in the same way. So you'd have to have that knowledge. But it's meant to be chemical specific. So you could take information, say that you understand, like if there's a mixture, right, multiple toxicants, as long as you understood those specific interactions, you might be able to say something about how broadly or how narrowly you would expect the effects to interact across taxonomic groups. So there's many ways you can utilize the tool, whether it's kind of chemical agnostic or very chemical specific. And if it, like I said, if it's multiple toxicants, you'd want to consider whether or not you know enough that you have enough evidence that they interact with the protein in the same way. And then you could start getting at that question. I think that's one of the things that maybe like with docking and being able to do some more virtual docking across many chemicals and understanding that interaction, you might be able to expedite that type of a process and understand multiple toxicants at one time more computationally. Could SeqAPASS be considered for a full day course at a coming meeting at such as CTAC? Absolutely, and we've done 2 courses at CTAC previously, and they've been relatively well attended. And so, absolutely, if there's a desire for courses, we have it together already. We did for CTAC Dublin, which ended up being a virtual course. We did a recorded training session, and so that was available through CTAC. But outside of those professional meetings, our team is interested in having the ideas and thoughts from other users, and so we're happy to, if it makes sense for a group or an organization that's interested in applying the tool, we're happy to do some training with individual groups as well as people reach out. Sounds like a plan. I tried to leave quite a bit of time for some questions, but if I don't see any more in the chat right now, I'll just bring you to the SeqAPASS page and just mention that, the login, you can request a login and everybody is granted a login. It's just to get, people to be able to have their own individual accounts and be able to keep track of the runs that they're working on individually. So it's not like your information or your data that you're generating is shared with any the other users. So like I mentioned, there is a user guide that is very useful, but I recommend getting training and then utilizing the SeqAPASS tool. It is much easier. We also have like information buttons throughout the tool to guide the user. Let's see. So Jess asks, do you have any plans to integrate sequence-based information in SeqAPASS with functional data? So RNA-seq reads. Primarily what we've been doing, because we want to take advantage of existing information and not necessarily rely on us generating data. So primarily we've been utilizing the whole back end of SeqAPASS takes advantage of the National Center for Biotechnology Information Databases and Tools. So NCBI, as most of us know it, out of the National Institutes of Health. They have their protein databases and their functional domain databases, et cetera. And so any of the sequence information that gets deposited in that public repository is it gets brought into the back end of SeqAPASS, so we can probe all of that data. And then we can also, we were talking about transparency as well. We also want to be able to link to the information that's available for those proteins within the NCBI database. So any sequence information that's generated and placed into these public repositories are available on SeqAPASS, which is really important because think about how much sequencing has advanced as far as reduced costs, better annotation methods. There's consortiums focusing on bringing in new whole genome sequences into these types of databases. So as those databases grow from public submission of those sequences, so too can our capabilities to extrapolate across more species. And so that's what the SeqAPASS tool utilizes is the NCBI databases and tools. As far as RNA-seq, so we have some projects in our lab using RNA-seq and looking at whole transcriptome to derive transcriptional point of departures for a variety of species. And so there's ideas there to be able to utilize SeqAPASS again. We'd be utilizing model organisms in those cases to generate the transcriptional point of departures to be able to extrapolate information to, say, endangered species or other species. So in that respect, that might be another way of answering that question as well. All right, so another question. In order to get a SeqAPASS login, I need your phone number or name, email, and phone number of a contact. So actually, let's see, I'm going to show that here. On the main login page of SeqAPASS, I'll show you, it gives you, it gives you the information that you need. So if you want an account, click here for instructions. And there it should give you the information that you need. So you go to this particular link and you click self-register. There's a form, so just like Marilyn mentioned here, is you have my contact as well as the email address as well as the phone number, and then you fill in that information and you select the SeqAPASS users community, and then that gives you access to the SeqAPASS tool. And I guess if anybody's having difficulty registering, or not registering, but getting your account, just reach out and we'll make sure that's it should be a pretty straightforward process. But if it's not, please reach out and we'll make sure that you get your account. All right, Sammy, I'm not seeing anybody typing and I don't see any additional questions.
Yeah, I think that if that's the case, we can just give everyone 10 minutes or so back.
Absolutely.
But
Yeah.
thanks so
So
much,
I hope
Carlie, for this
everybody
great
gives it a
presentation.
try, and again, reach out if you have questions or are interested in some additional training.
All right, great. Well, thanks everyone for coming. And just some quick housekeeping. So this is our only presentation for December. We will not see all of y'all until next year in January. And then I'm excited to see you then. Hope everyone has a
Thank
great
you.
rest of their year.